Good morning. Good morning, everyone. Happy Wednesday. Good morning. All right, we're still just a minute before 9 o'clock, so we'll let some people come on in. If you would, go ahead and type your first name or your nickname into chat for me. Thank you. Good morning. Good morning, everybody. I hope you all had a nice Memorial Day weekend. Very good. Thank you. All right. We're still looking for a couple of folks. We'll give them a minute to come in. So what we're going to be doing today is having our next laboratory exercise. And today we're going to be talking about how we examine living microbes underneath the microscope, because there are certain techniques that we use specifically when we want to examine a microbe that is still living. So sometimes we want to examine samples of material and we're curious whether or not there are certain microbes in that sample. And so we use particular techniques to do that. And that's what we'll be talking about today. Before I begin our exercise, though, I just want to take care of a couple of housekeeping things. First, I wanted to say thank you to everybody who got all their work in during the first week of class. I was really pleased to see that. We had 2 quizzes and we had lab questions and everybody got their work in, which is wonderful. Keep it up. For the most part, everything was well done. Quizzes and lab questions, well done. You probably noticed, if you've looked at the work that you submitted, since I graded it, if you've looked, you've probably noticed that I leave you comments on your work quite a bit. If you get a question wrong on a lecture quiz or on laboratory homework questions, what I usually will do for you is leave you a comment. And I'll explain to you what you missed or why your answer was wrong or why your answer might have been incomplete. If it's a multiple choice question, I usually don't leave a comment, because on the multiple choice questions, you're shown the correct answer. But if it's a short answer question, an essay type question, you can expect me to give you a little comment underneath the question. So when you open these assignments up after I've graded them, there should be somewhere on Canvas that says something like view instructor comments. There should be a little link that you can click on associated with each assignment. And that will allow you to see my comments. So again, I was really pleased. You may have had a little bit more trouble with the lab questions than the lecture quizzes. The lab questions that we have in this course, some of them can be quite challenging. There's usually one question at least on each laboratory assignment that is quite challenging. It's usually the question that's listed as a clinical application. In other words, there will be a question that is very, very practical, related to the clinical workings of microbiology, the actually practical applications of microbiology. So there'll be at least one question on each lab homework that I suspect will be quite challenging for you. There is one thing to remember, though, when you answer a difficult question like that. If I've asked the question, I've told you the answer. So you don't ever have to go searching for the answer. In fact, you shouldn't. You shouldn't be Googling any of these questions. You should be using your notes, because I won't ask a question that I haven't told you the answer to. It's just that you may not have caught it in lecture. You may not have thought it was an important thing to write down. Yeah. So on those clinical application questions, always think very practically. In other words, don't overthink it. Think in very practical terms. For example, one of the questions last week's assignment was about a person who was suffering with pneumonia, and we obtained some sample material, some sputum from that person, and we examined it under the microscope, and we saw a microbe. We saw a microbial cell under the 100x lens. And the question was, how do we know, or why would we immediately rule out archaea as the type of organism that this was? And the answer to that question is because archaea don't cause disease in humans. You see what I'm saying? It's a very practical question. If I'm looking at a sample from a person who's suffering with a disease, and I see a microbe in there, I can automatically rule out archaea. Automatically. And that's because the archaea don't cause disease in humans. Now, a lot of you, well, not a lot of you, but a few of you told me that the reason it couldn't be archaea is because the archaea live in extreme environments. And we wouldn't consider the lungs of a human being to be an extreme environment. That was a very reasonable answer to that question. Wasn't what I was looking for, but be sure that in your notes, you have written down somewhere that yes, the archaea can be found in some very extreme places. We talk about that in the lecture, but that doesn't mean they're only found in extreme places. The archaea can also be found all throughout the environment. They can also be found, importantly, in our human microbiome. The most current research suggests that about as many as 7% of all of the microbes that live on us and in us are archaea. We don't know enough about archaea yet. We don't know as many individual species of archaea as we do of bacteria. Really, we're only in the infancy of understanding archaea. But yes, you can find archaea on and in your body. You can also find viruses in the human microbiome. Remember, the organisms that are in your microbiome are either completely harmless to you, they just sort of live on you or in you, or they're helpful to you. They help keep you healthy. Bacteria, archaea, viral particles, certain kinds of yeast, which is a fungus, they all live on us. Remember, the estimate is something like 30 trillion microbial cells are on you or in you right now. Many, many more microbial cells are on you than human cells. You are mostly microbes. And yes, the archaea are part of that collection, interestingly enough. So yes, the second part of that clinical application asked you why it also couldn't be a virus. We know it's not an archaea, and we know it's not a virus. And the reason we know it's not a virus is because we saw it under the microscope. We saw the pathogen, the microbe that causes this pneumonia. We saw it under the microscope. It can't be a virus because we can't see viruses under the microscope. So again, automatically, I know that. If I see a microbial cell under there, I know it's not an archaea, and I know it's not a virus. In all likelihood, it's a bacterium. Bacteria and viruses are the two most common pathogens in humans, bacteria and viruses. And since I can see this microbe with my microscope, it's a bacterium. I do want to point out too, we don't talk, we talk a little bit about human disease in this course, but a couple of you told me that viruses don't cause pneumonia, or that, in other words, it couldn't be a virus because viruses don't cause pneumonia. Again, we don't go into great detail about human disease in this course, but again, bacteria and viruses are the two big groups of pathogens in humans. And yes, viruses can cause pneumonia all by themselves. The pandemic virus that we've all been living with, SARS-CoV-2, when it originally emerged as a pandemic virus, as a new or novel pandemic virus, the primary symptom that it was causing in people was a pneumonia. So in the early days of the pandemic, the very first strain of that particular virus caused a very severe pneumonia in people. And if you remember back in, gosh, March and April of 2020, there was a great concern about having enough ventilators. Do you remember? We were worried that we wouldn't have enough ventilators in our hospitals for all of these people that were coming in with a viral pneumonia. Pneumonia is a disorder where fluid builds up in the lungs. And obviously it interferes with your ability to breathe and get oxygen into your blood. So yes, just so you know, just so you can put it in your vault of knowledge, viruses can absolutely cause pneumonia in humans, as can bacteria. All right. Now, I do want to pull up one slide for us just very briefly. Hold on just a sec. This is a slide from the very first lecture, the Intro to the Microbes lecture. And this is a slide that we went through in the lecture talking about the relative size of microbes. If you haven't already, I want you to put a star next to this. I want you to make sure that this information is in your lecture notes, because that's how important this is to us. It has very practical importance for us in the laboratory because it reminds us what we can and cannot see under a microscope. Remember, plant cells and animal cells, these are eukaryotic cells. And they're going to be the biggest cells that we would ever see under a microscope. Bacteria are the limit, the smallest thing that we can see with a light microscope. If you go below that size, if you get down into the nanometer range of size, viruses, for example, you would need an electron microscope to see those things. You can see all types of cells with an electron microscope. It's just that you need an electron microscope to see a virus. You cannot see a virus under a light microscope. The other thing to know is this, and we'll take a look at some bacteria today under the microscope. Bacteria are at the limit of what we can see. If you look at the bar down here for the light microscope, we are right at the limit of what we can see with a microscope when we're looking at bacteria. Now, why is that important? Well, that means that when we look at bacteria under a light microscope, we are not going to be able to see them in great detail because they are the very smallest thing that we can see with a light microscope. We're not going to be able to see individual structures on a bacterial cell under a light microscope. In other words, you're not going to be able to tell whether you're looking at a peptidoglycan cell wall or an S layer under a light microscope. You simply will not be able to tell that kind of detail. You won't be able to see the genetic material or the ribosomes in that cell. All you'll be able to see is the relative size and the shape of that cell. And again, we'll take a look at some bacterial cells today so you can see exactly what I mean. You can see bacteria under a light microscope, but you cannot see fine structural detail. All right, very good. Does anybody have any questions about any of the work that you did for me last week? Any of the lecture material you watched? Feel free to type it into the chat if you do. Remember, if you ever have any questions during a lab period, just type it right out into the chat and I'll see it there. Okay, so if you haven't yet, go back and look at your lecture quizzes from last week and look at your lab homework from last week. See what you might have gotten wrong. Read my comments. And if you need to, go back into your notes and update your notes. Okay, if you don't have it in your notes, put it in your notes. If you had it incorrectly in your notes, fix it. Okay, because remember, your notes are what you're going to be memorizing for the lecture exam. All right. All right, let's pull up our homepage on Canvas. We're actually looking at the modules on our Canvas page. I'm going to scroll down into week 2 right here. I'm going to open up our to-do list for the week. So remember, you have one lecture to take care of this week, and that's the cell. The lecture quiz is due by midnight tonight. The first lecture exam takes place this week. It covers all the lecture topics that we've covered so far. So the introduction to microbiology, history of microbiology and microscopy, and the cell. So these are the 3 topics that will be on the exam. The exam is available to you starting at 8 o'clock tomorrow morning. You can pick when you want to take it. You just have to get it into me before Saturday at midnight for full credit. Okay, remember, this is a closed note, closed book exam. And you have to go to the exam through the lockdown browser. In other words, you open up the lockdown browser, and then you go to Canvas, and then you open up the exam and take it. If you try to go to the exam in, say, Firefox or Safari or something else, you'll get a message that says, sorry, you have to take this exam using the lockdown browser. You'll have 90 minutes to complete the exam once you begin. You can take longer than that. In other words, the exam isn't going to suddenly close on you at 90 minutes, but you can only use 90 minutes if you want to get full credit on the exam. Similarly, if you submit the exam late after Saturday at midnight, you'll also lose some points for that. Okay. Just remember too, you have a lab homework assignment this week. It says microbes in the environment. It should actually say examining living microbes. That's what our lab is called today. But yes, you have a lab assignment this week too. So make sure it's all on your personal calendar so you don't forget anything. All right, I'm going to go ahead and open up our lab exercise. Remember, these slides are available to you on Canvas. So you certainly do not have to write down every single thing that you see in this slide set. You don't have to do that. If I were taking notes during a laboratory like this, what I would do is I would take notes by slide number. Slide number 1. That's just a title slide, so no need to take notes. Then I might write slide number 2. And I would write down any other things that are not on the slide. That's what I would put in my notebook. And then when I was ready to study, I would pull the slides up. I would open up the slide set and use the slides while I studied. That way, you're not spending a whole lot of time and energy during the lab trying to write down every single thing, everything that's on the slide, plus everything that I'm saying. Do yourself a favor. In other words, don't try to write down everything that's on the slide. Just write down any extra things that I'm saying. You already have the slides. You don't need to write them down. Okay? All right. So today we're looking at living microbes. We have 2 objectives today. Number 1, we're gonna begin by talking about one of the functions in a light microscope called phase contrast. Remember, when we use a light microscope, we can choose what kind of functionality we want. We can choose the bright field microscopy, we can choose dark field, and we can choose phase contrast. And today we'll talk a little bit about why we would want to use phase contrast when we're examining living microbes. We'll talk about how you make a type of slide called a wet mount slide. We'll talk about how you make a hanging drop slide. And we'll talk a little bit about motility today. We'll talk about how you can recognize when a microbial cell has motility. In other words, when it's able to move under its own motion. And we're going to distinguish that true motility from what's called Brownian motion, which is sort of a false motility that we sometimes see underneath the microscope. In the second part of the lab, what we'll do is we'll actually make a wet mount and we'll examine it underneath the microscope together. So let's start off first by talking about when we would bother making some kind of a slide preparation that allows us to look at living microbes. You have to understand that most of the time in the microbiology laboratory, when we're examining microbes under the microscope, we're going to stain them. We're going to make a preparation where we put the microbes onto the slide, we affix them to the slide, we attach them to the slide, and then we stain them. And the reason we put stain on a microscope slide like that is because it allows us to see the cells better. It allows for better visualization using either the bright field function of the microscope or the dark field function. Either one of those works very well with stained cells. Now, here's the thing though, if you apply a stain to a microscope slide with microbes on it, if you apply a stain, that's going to kill the cells. Now, remember, part of what we do is we attach the cells, we fix the cells to the slide. That process alone will kill cells. And then we apply a stain to the cells, and stains are toxic. Microbiology stains are toxic. So if we're going to stain a cell or a group of cells, in order to examine them, we're going to kill those cells. Now, there are times when we want to look at a sample of material without killing the cells. There are times when we want the cells to be alive because we're interested in learning if there are any microbes in that sample that are motile, that are able to move on their own. So we can't kill them or they won't be moving for us. Now, when would that apply? When would we be looking to see if there are any mobile microbes? Environmental samples is the answer. For example, in the microbiology lab, we're often asked to look at routine water samples from recreational bodies of water. So for example, in the state laboratory here in New Hampshire, the one that's found in Concord, recreational parks, if you will, send in samples periodically, particularly through the summer months, and they ask us to examine them looking for microbes because people are going to be swimming in these lakes and swimming and fishing in these rivers and so on. So that kind of work is done routinely. We monitor those bodies of water and make sure there are no high levels of dangerous microbes in there. We also examine samples from water treatment plants. Water treatment plants are trying to get water back into the environment after it's been treated to get rid of microbes that come from human waste and so on. Water tanks and cisterns. There are a lot of places here in New Hampshire where people are drawing their drinking water out of a well. Some people are storing water in tanks or cisterns. If you've ever been to New York City or other large urban areas, you know that there are a lot of apartment buildings, tall apartment buildings that use water tanks as their water supply. That water has to be monitored for the presence of microbes. Even things like humidifiers, industrial locations, commercial locations that have elaborate ventilation systems. If you can imagine those big office buildings that run elaborate 24-hour ventilation systems, those have to be monitored because there are certain microbes that can grow in ducts in ventilation systems. So they have to be monitored to make sure that there's nothing dangerous growing in them. So these are the times that we don't immediately stain the samples. We instead examine them first without any stain because we're looking to see if there's anything in there that is swimming around, any microbes in there that are moving under their own power. Now, on the light microscope, we're going to choose phase contrast. That's the functionality that we're going to choose when we're looking at these living samples. And the reason we choose phase contrast is because it allows us to get the best view. Pardon me, I'm just trying to enlarge our view here. It allows us to get the best view of the sample. Whoops, that's a little too large. Take a look at the top of this slide on your screen. Over here on the right, this is a sample of cells under the 20x objective lens. Now, these don't happen to be microbial cells. These are actually animal cells. This is a sample of animal cells underneath the phase contrast portion of the microscope. I use this image because I want you to notice that some of these cells have a halo around them, a white halo. What phase contrast does for us is it allows us to take advantage of the refractive properties of three-dimensional living cells. you'll notice that some of these cells are refracting the light, and we're getting sort of a shine off of the cells, essentially. Now, if you look over here on the left, this is the same sample of animal cells, but now we're under the 40x lens. And again, you can see this circle of white around some of these cells. In fact, if you look, all of these cells have a little bit of sort of shine around them. Can you see that? All of these cells have a little bit of white around the periphery. This is what I mean by phase contrast allowing us to see refractive properties. It helps us. It helps us see the cell in three-dimensional detail. Now, here's another example of why we need to know the relative size of things when we work with a microscope. I told you that those two slides were animal cells, but if I hadn't told you that, you should be able to tell me that now. Those slides are clearly, those cells are clearly visible under 20x and under 40x. Under the 40x, they're actually big, right? They're big cells. That's how you know they're not bacteria. We can see bacteria under a 40x lens, but they're very small. You really have to go to the 100x oil immersion lens in order to see bacteria in any kind of detail. Let's look at this slide again. These cells shown in black, all of these black structures in here, these cells are under the 20x lens. We cannot see bacteria under a 20x lens. I can see these cells. That tells me they're not bacteria. Under the 40x lens, these black cells are big. Not only can I see them, but they're big. That tells me they're not bacteria. Okay, so always be thinking in your head, what magnification am I under here? And what can I see at that magnification? Can I see bacteria under that kind of lens or no? And that will give you a hint as to what you're looking at, especially when you get a sample from a real world environment, like a lake or a water tank or a ventilation system. Always be thinking, what could this be under this magnification? So phase contrast allows us to see small differences in refractive properties of cells. And that shows up as differences in brightness. And that gives us a little more detail than we would get without phase contrast. On this slide right here, you're looking at a couple of samples that came out of a body of water, a recreational body of water. Now, this particular sample, these samples, were centrifuged. They were put into a centrifuge to gather all of the material together in a pellet. And then that pellet was essentially put onto a microscope slide. That's why there are so many things visible all together. Normally, we expect environmental samples, water samples primarily. Normally, we expect them to be quite dilute. We don't expect to see very many slides per drop of water, or sorry, very many cells per drop of water. So the fact that you can see lots and lots of different cells here, that tells you that this sample was put into the centrifuge in order to gather all the little bugs and bits together so we could look at them. Now, one of the things you notice over here on the right is that I've got all kinds of things in here, don't I? I've got literally little animals in here. I've got some very large cells. Now, this particular slide, This slide has been colorized. That's why you're seeing things in different colors here. But this particular slide, this view was taken under a 40x lens. So if you look, you can see some very small little black dots on this slide. Those could be bacteria. Those little black dots could be bacteria. That's how big bacteria are under a 40x lens. This brown circle is much too big to be a bacterial cell. This blue elongated structure, that's much too big. This thing right here in brown, that's much too big to be a bacterial cell. Only these little black dots could be bacteria in that image. If you look over here at this slide on the left, same thing. I've got some plant cells in here. I've got some algae in here. I've got some diatoms in here. I've got all kinds of interesting little creatures in here. Again, this is from a pond. And we expect ponds to be just teeming with microbial life. When I look around on this slide, I can just barely make out some very little tiny black dots. Just the tiniest little dots in the background of this slide. You may not be able to see them, but if you look up close, there are some tiny little black dots. Those could be bacteria. The rest of these are eukaryotic cells. And I know that because I'm only under, this is either I think this is a 20x lens or a 40x. It's one of the two. All of these larger things, they cannot be bacteria. They're just too big. Any questions? Any questions so far? You can imagine that if it's your job in the laboratory to examine a sample from a body of water and let whoever sent the sample, let them know whether or not there are dangerous levels of microbes growing, you have to know which microbe is which. And again, most of the dangerous organisms for us that you would find in a body of water, most of them are bacteria. If you've ever been to a lake or a pond in New Hampshire, even to the shore, frankly, in the middle of summer, you may have seen a sign posted at the beach saying that the water is not safe right now and that you need to stay out. What that means is that a routine sample was taken and the number of bacteria in that sample was just too high. For whatever reason, there are too many bacteria growing in the water right now. And it's too dangerous for you to go swimming in there because you might accidentally swallow some of that lake water. Austin's asking a great question. Are there any unusually large bacteria that can be confused with animal cells? Austin, the answer is there are some bacteria that are particularly large. And even more interesting is that some bacteria group together. They live in sort of communities together. And depending on the species of bacteria, they might actually join together in a chain, or they might join together in a cluster, like a cluster of grapes. Those are very visible under the microscope. And I guess in theory, you could confuse those with a larger cell. That's a great question. As you probably have guessed by now, a lot of the work that we do, a lot of the ability for us to determine what we're looking at in a sample comes from experience. We learn what kind of microbes tend to be found in freshwater lakes or in rivers and streams, or at the shore in brackish water. It takes a lot of experience, but you start to learn what you're likely to find in terms of bacteria in those places. Does anybody know the scientific name of the organism that causes us the most problems in freshwater swimming places like lakes and streams? Type it into chat if you do. I'll tell you, it's the same organism that is sometimes found in drinking water. Oh, Amy's guess is cyanobacteria. Amy, that's a great guess. And yes, you're right. Cyanobacteria are a huge problem for us here in New Hampshire and in other parts of the country. But I was thinking of another bug. Brooks got it. E. coli. One of the biggest reasons that a beach, for example, at a lake, a beach would get shut down is because there's been an E. coli sort of bloom in the water. E. coli is an organism that lives in the intestine. So it could be from human fecal material in the water. It could be from the birds that are floating around, the ducks and geese on the lake. It could be from any of a number of animals that live around the lake and might actually defecate into the lake. That's where E. coli comes from the intestines of animals. And sometimes it tends to grow to large numbers right at the shore, right at the beach. Yeah, right where the water is shallow, right where the water warms up a lot. Yeah, right where the little kids tend to splash around in the water. Yeah, E. coli can be a real problem for us in recreational bodies of water. The good news is if some kind of a bloom event happens and a beach has to be closed down, it's usually only for a few days because nature will take care of that for us. When a bacterium grows to large numbers in a body of water like that, what will happen is it will eventually outgrow itself. All that E. coli that's growing will run out of nutrients and run out of oxygen and die. So it's sort of a self-limiting problem. Now, cyanobacteria are different. Cyanobacteria are environmental. In other words, they don't come from our bodies. They live in lakes and streams normally and ponds and so on. But because of runoff, because of the kind of man-made chemicals that end up in our recreational bodies of water, they also can grow out of control at certain times of year. And the thing about cyanobacteria, that name cyano means blue or blue-green. You've probably driven past a body of water in, like, let's say August, and you've seen a green slick near the shore. That's probably cyanobacteria. It looks a lot like algae. In fact, we used to call cyanobacteria blue-green algae. But once we developed more sophisticated techniques in the laboratory, we realized that this organism was not an algae, but a type of bacteria, a photosynthetic bacterium. So cyanobacteria will bloom into a big green slick, but just like I said a few minutes ago, they tend to outgrow themselves. There are so many of them in that one area that they run out of food. They tend to run out of oxygen and they die. So if a beach gets closed down for a cyanobacteria bloom, it will tend to open up again within a few days. Very good. Good answers. The reason cyanobacteria are dangerous to us is because when they die and the cell falls apart, they release toxins into the water. Every year there are stories. It's usually a dog, sadly. It's usually a dog who goes running into a lake and goes right through the middle of one of those green slicks and comes out of the water sick. Yeah. And that's because, again, toxins get released into the water, and they're dangerous. They're dangerous to animals. Very good. Cyanobacteria have become more and more of a problem for us here in New Hampshire because our waters are polluted and climate change. Climate change is warming the water. Also problematic. Amy's asking, is cyanobacteria respiratory? So cyanobacteria is not a pathogen normally. In other words, it doesn't grow in us normally. It's an environmental bacteria. It lives out in the environment, in the water. What makes it dangerous to us is when we get exposed to the toxin that it releases in the water. There are two types of toxin that cyanobacteria make. One of them is a liver toxin, and one of them is a neurotoxin. And there have been people who've died swimming in lakes that have cyanobacteria blooms. There have been people who've died from it because of those toxic things. Good questions. All right. So I said at the beginning of the lab that there are two types of slides that we can prepare to examine a water sample and look for motile bacteria. Look for bacteria that are moving or swimming. The first one is called a wet mount, and the second one is called a hanging drop. Now, the reason we would make a wet mount is in order to see, first of all, just if there are any bacteria in there, but also to see, again, if they're moving around. Are they swimming around? What you're looking at on this slide is a sample of bacteria in a wet mount. Now, this is a short video. I'm going to turn it on for us in just a minute. You can just see the bacteria here. Can you see them? We're going to look at this view, which is 40x, under the 40x lens. And then we're going to look at the same sample under a 100x lens. So the reason that this looks blue is not because it's stained. I know that's confusing, but this is actually a freshwater sample. And in order to take the video, what they did was they used a filter. If you remember when you were learning about the microscope from our little tutorial handout, we said that on the substage assembly, where the diaphragm is and the condenser is, there's also a place to put filter discs. There's a holder for filter discs. These are just glass discs that are colorized. So you can put a little disc in there with a piece of colored glass, and it will make your sample look that color. So what they did here was they used a blue filter, just so we could see it a little bit easier. Let's put this in motion. There are actually two kinds of organisms in here. One of them is called Enterobacter, and one is called Rhodobacter. If you look down at the bottom here in the notes section, you can see I've written out the whole name of these two organisms. Not that it's important for you to learn these or anything. I just want to introduce you to some of the things we sometimes find in water. Enterobacter aerogenes, that's one. And Rhodobacter spheroids, that's the other one. All right. Now take a look under the 40x lens. Can you see the motion? These bacteria are swimming. Now I've moved up to the 100x lens, so I have more detail. Look at those little buggers swimming around. Now you'll notice that some of these cells look dark, look black, but some of them look white. Can you see that? The reason for that is this. When we make a wet mount, we are literally putting a drop of liquid onto the slide. So when we look through the drop, there's going to be different planes of focus. Some of the cells are way down at the bottom of the drop. Some of them are in the middle of the drop. Some of them are at the top of the drop. And depending on which plane that we focus in on, the cells are going to look different to us. So you can clearly see at 100x that these cells are moving around. These cells are motile. Both Enterobacter and Rhodobacter, this one is Rhodobacter, are swimmers. They are motile. Now look at this, there's a clump of cells here that's not moving. Here's another clump over here. You're always going to have some cells under the microscope that are dead. And that's what you're seeing here. Remember, these cells are not stained. We haven't put any stain on this. What we've done is we've used a blue filter, and it helps us see the cells a little more clearly. This is 40x. This is 100x. You see what I mean? How you get much better detail under the 100x lens? Remember, if we're using the 100x lens, we have 1000x total magnification. You take the power of the objective lens and you multiply it by 10, because that's the magnifying power of the eyepieces. We're also under the 100x lens here, and we can see that these cells are swimming around. They are motile organisms. Now, on this slide, we have a different sample. This happens to be an organism called Listeria Monocytogenes. Again, you don't need to know this name. I'm not going to test you on this name, but I'm going to introduce you to all different kinds of bacteria through the summer. Listeria Monocytogenes is a pathogen. And this happens to be one that we find in food quite a bit. It loves to grow in dairy products, especially soft dairy products like soft cheese. Listeria has a very characteristic type of motility. We call it tumbling motility. Let's take a look. First of all, you can definitely tell that these cells are moving, but I want you to try to follow one cell. Look at how it's moving like in a circle almost. It's flipping over as it moves. Try to focus on one cell and notice how it flips around as it moves. This is a certain kind of motility that we refer to as tumbling motility. They tumble around as they swim. Now, again, notice the different planes of focus. Some of the cells are clearly visible. Those are the black ones. Some of them are not in focus. Those are the white ones. See those? Those are at a different plane of focus. If I were to adjust the fine focus knob on my microscope, I could bring these white cells into focus and they would look black. All right, let's look again. The other interesting thing about Listeria is it likes to form short chains of cells. Here's one Listeria cell in black. Now look at this one over here. There's two of them end to end attached to each other in a chain. Let me run this again. Here's a chain here. Here's a chain right there. Look how long that one is. Here's a chain right here. A lot of bacteria does this. Here's a chain. A lot of bacteria prefers to live in groups. They take on certain formations out in the real world. Some bacteria like to stay as individual cells, but other ones will group together. And Listeria likes to form chains. Now, Listeria causes a disease called listeriosis, and it's extremely serious in pregnant women. Adults tend to recover from listeriosis with antibiotic therapy, but the fetus is much more severely affected. And that's why when women are pregnant, they are warned by their obstetrician to stay away from soft cheeses, things like brie or soft sort of melty looking cheeses. I don't tend to eat those cheeses anyway, but if you're a pregnant woman, you will be asked to just refrain from eating those cheeses just in case they happen to be contaminated with this organism. Now you can find Listeria in other sources. Again, it particularly likes dairy. There have been cases where Listeria has been found in ice cream. There have been recalls of batches of ice cream that are contaminated with Listeria. And again, treatable with antibiotics, but very dangerous to the developing fetus. Okay. So let's talk for just a minute about how you make a wet mount and how you make this other kind of slide called a hanging drop slide. These are two different types of slides that we would prepare. And the purpose for making a wet mount, the purpose for making a hanging drop is because we're looking at living cells and we're trying to figure out if they're motile. Wet mounts are very, very common. We use them a lot, and that's because they're very easy to make. They'll allow us to examine living organisms that may or may not be motile. That's why I have the plus or minus here. We use wet mounts to examine living organisms that may or may not, plus or minus, be motile. Hanging drops are a little harder to make, and so we don't use them as much. We would use a hanging drop specifically for motility. We're trying to find motility. Regardless of whether you're making a wet mount or a hanging drop, you've got to be sure that that sample of water is very well mixed. You can imagine, like I said earlier, a lot of these samples are very dilute. And what you wouldn't want to happen is have the cells settle out to the bottom of the test tube or whatever container the sample is in. You wouldn't want the samples to settle out to the bottom and you draw just plain water and put it on your slide. So we always make sure to mix up these water samples very thoroughly before we draw out a little bit and put it on a wet mount. We're going to use either a sterile pipette or an inoculating loop. And I have a short video for us to look at using an inoculating loop. That's going to allow us to take a little bit of that sample out and make our wet mount. We're going to mix up the sample thoroughly, and then we're going to draw out a little bit from the center of the sample. In other words, if I got sent a test tube and it has 20 mLs of water in it, I'm going to mix that up. And then I'm going to take my pipette or my loop, and I'm going to get a sample from the center of that volume. So if the volume is, let me get my hands correct here. If it looks like this, if this is the top and this is the bottom of the volume, I'm going to draw from right in the middle. If you do that every single time you make a wet mount, you're more likely to get a representative sample from your environmental water. We're going to take a microscope slide, labeled, of course, like we talked about last time we were together. We always label everything in the microbiology lab. And we're going to put a drop of water right in the middle of the slide. So the middle third of the slide, right in the center. We're going to put a nice big drop. That is a wet mount. That's what a wet mount is. Okay. Super simple to prepare. Very helpful to us when we're trying to look for living organisms that may or may not be mobile. What you're looking at on this slide are those preparatory steps. Here's a test tube. You can see the sample inside is colored blue, just so we can see it a little more easily. And the person is putting that test tube in a vortexer. That vortexer is going to send like a little tornado of vibration up through that tube, mixing the sample really well for us. We're going to take a sterile, single-use plastic pipetter. This is called a transfer pipette. Has a little bulb on the end so we can suction up some of the sample. And we're going to want to take our sample from the center of the volume. Over here, you can see some volume in a beaker. So this sample came in a beaker. And you'll notice that the person's putting the tip of the pipette right in the center, right in the middle of the volume in the beaker. If there's 50 mLs in here, I want to draw my sample from about the 25 ml mark. I'm going to place my droplet of water right on top of my slide. By the way, here's a little trivia for you. A nice sized drop of water has about 20 microliters in it. 20 microliters makes a nice sized drop. So if you're using a transfer pipette, you literally just put a drop on the slide. But sometimes in laboratories, we're using micro pipetters where you actually set the volume that you want to draw up. So if you're looking to draw up a drop's worth, you'll want to draw up about 20 microliters of fluid. Now, here's the cover slip that's going to go down over my water droplet. Here's the slide. We're looking at the slide from the side. So here's the water droplet. And what I'm going to do is I'm going to put one edge of the cover slip down, and then I'm slowly going to lower the cover slip down to the slide so that I can examine my water droplet. Very straightforward. Now, there are some limitations to wet mounts. The biggest limitation is this one. When you put a drop of fluid, water, onto a slide and put a cover slip on it, and then take it to the microscope and examine it using light, using plain old visible light, that light is going to evaporate your sample pretty quickly. It's going to warm it up and it's going to start to dry. It's going to start to evaporate. You have about 15 minutes to look at your wet mount. Now, let's say you're examining a wet mount and you really want it to last a little bit longer than 15 minutes. There is a trick that we use, and that is we take a little petroleum jelly and we put it around the edges of the cover slip. You can do this using a toothpick, for example. And then when you put your cover slip on, you've created a little bit of a seal around the edge. And that will allow that wet drop to last a little, that wet mount to last just a little bit longer. You have to remember though, the light source on a microscope gets very hot and it will heat up that liquid and it will evaporate it pretty quickly. So that's really the only limitation to a wet mount, is that it's not going to last very long. So if you're just looking to see if there are cells and if they're motile, you can do that within 15 minutes. That's not too hard. Remember, on a wet mount, you won't be able to know what kind of bacteria you're looking at. You won't know what its genus and species is. You'll simply know that there are bacteria in that sample and that they're motile or not. That's the information that you'll get from looking at that. All right. We're going to take a little break here. It's just about 5 minutes after 10. So we'll see you back here at 10 after 10. Go get yourself a cup of coffee, run to the restroom, stretch your legs, and we'll be back in 5 minutes, okay? Okay. As you come on back in, go ahead and type your name into the chat for me. Quick attendance, thank you. Very good. Ooh, one of my eyes is bothering me. I'm having the worst time with the pollen this year. I don't know about you, but the pollen really bugs my eyes. There were some amazing photographs on the news last night that were taken down in Massachusetts. They had a strange weather event yesterday where the temperature dropped 20 degrees in about 15 or 20 minutes. And that's pretty bizarre all by itself. But the reason it was able to drop so far so fast is because sort of a windstorm came in and a whole bunch of pollen got released into the air. And there were photographs on the news of this green haze in the air in the Boston area. And all I could do was just shudder from thinking about all that pollen. What are you going to do? It's that time of year, I'm afraid. All right, if you haven't already, type your name into chat for me. And we're going to jump right back in where we left off. And that's talking about the second technique, which is called a hanging drop preparation or a hanging drop slide. Now, the difference between a wet mount and a hanging drop is essentially that in a hanging drop slide, instead of putting the drop down onto the slide, we're going to hang it from the cover slip. On a wet mount, we put the drop on the slide, but on a hanging drop, we're going to hang the drop from the cover slip. And it's going to allow us, again, to look at living cells and see whether or not they're motile. It's especially useful when you're looking, again, at these environmental samples. Now, when you want to make a hanging drop, you cannot use a plain old flat microscope. You need what's called a depression slide. I'll show you a picture of one of these in a minute. But what a depression slide is a glass slide that has a little depression in the middle of it. It has a little cup carved out in the middle of it. You also need a cover slip, of course, and that's what we're going to hang the drop from. And you need some petroleum jelly because we are going to secure or sort of seal down the cover slip over that depression in the slide. Down here, you're just looking at, I think it's a branch from a rose bush after a rainstorm, and you can see this droplet of water hanging down. That's what we're trying to create. We're trying to create a droplet hanging down from a cover slip into that depression, that well on the slide. On this slide, we're looking at a depression slide over here on the right. It looks like a regular microscope slide, except it's got this circular depression cut into it. So this would be concave. And if you were to put your thumb on it, it would be a depression or a well cut out of the glass. When you look at a depression slide from the side, you can see that cutout area right there. So what we're going to try to do is hang a drop. We don't want the drop to touch the slide because we want it to stay as a drop. So we hang it down into that well or that depression without allowing it to touch the depression. And we're going to hang it off of a cover slip. The little beads that you see between the slide and the cover slip, those represent petroleum jelly. We're going to want to seal that cover slip down so it doesn't move. Now, hanging drops are a little tricky to make, as I'm sure you can imagine, because if your drop is too big, what's going to happen is when you hang it upside down from the cover slip, it's going to hit the glass slide, and the glass slide is going to essentially pull the drop down. You have to have a small enough drop so that it will hang down from the cover slip without touching the slide. They're tricky. Okay, they're tricky. Most people would rather make a wet mount because a wet mount is much easier and much quicker. But there are times when it's just hard. It's hard to see what's happening on a wet mount. And so you might use a hanging drop instead. Now, when you look under the microscope at a hanging drop, you're going to see something that looks like this. Now, I know this looks very strange, but what you're going to see is the drop right here is the edge of the drop. And the drop will be pulled over to one side. All of the cells that are in that drop are going to sink to the bottom. And that's where we're looking at them. That's where we're seeing them. We're actually examining the bottom of the drop. So this circle right here represents what you're going to see under the microscope. This line that twists and turns, this is the edge of that drop. And all the cells will be located between the edge of the drop here and the edge of your field of view. Most of the cells will be near the edge of the drop. If you look over here on this image on the right, this is an actual view. Now we're only looking at part of the edge here, but this is the edge of the drop. Here's the rest of the field of view. So this is the droplet over here. This is the edge of it, and that's where we're going to see most cells, right near the edge. Most of the cells are going to sort of accumulate right near the edge of that drop. And if they're swimming around, you're going to be able to see it. You're going to be able to tell. So all of these little black dots in here, the gray dots that you're seeing, those are cells, bacterial cells. And again, they're going to be drawn to the edge, that twisting, turning edge of that drop. Let's take a quick look at a little video clip of a person making 1, 2, 3 different hanging drops. Now, I didn't make this video clip. This came off of the internet, but I wanted to show it to you because it demonstrates pretty nicely how you do this technique. You'll see that there's material on her glass slides. There's sort of a translucent material here. There's a white material here. Over here, there's no material. I think this demonstration was filmed to show that you can use different sort of sealants around the cover slip. I think that was the purpose of the demo. Here are the cover slips. You'll notice there's one cover slip set out next to each slide. This is a test tube that has some material in it. This looks like culture material to me, not a water sample. This black device here that this person is sticking into the test tube, this is an inoculation loop. You'll be able to see it a little more clearly in the video. This person is working in a biological hood. I do see one thing that's very, very wrong here. Can anybody tell me what is wrong in this picture? Right away. Take a look at that person's hands. What do you see? Candy's got it. Maddie's got it. Casey's got it. There's no gloves. This person is holding on to a culture tube with bacteria in it, and there's no gloves. Very bad form. Very, very bad form. Now, again, I don't know where the video was originally made. It could be that they know that there's no bacteria in that tube. But remember, if you're handling a culture container, if you're handling any sort of container in the laboratory that has microbes in it, you have to have gloves on. All right, let's run this video and take a look. Remember, one of the big differences between a wet mount and a hanging drop is where we're going to put our drop of material. In a wet mount, we would put the drop right on the center of the glass slide. But in a hanging drop, we're going to put the drop on the cover slip. All right, let's take a look. So here's the loop. Notice when she comes down, she's going to put a loop full of this liquid onto each cover slip. See that little loop, that little wire loop? That's an inoculating loop. She's pulling up just the tiniest little amount of liquid with that loop, a tiny drop, and putting it right in the middle of the cover slip. She's going to put one drop on each cover slip. Then she's going to take the slide. She's going to flip it over and put it on the cover slip. She just flamed her loop. Again, very, very nice, because that loop now has bacteria on it. She put it into the Bunsen burner to sterilize it. She's putting a little bit of petroleum jelly down on these circular areas that she's put on the slides. Again, I'm not really sure why she used different material on the slide. But she's putting the petroleum jelly down on the slide because she's going to want to stick the slide to the cover slip. See the depression, the well in the slide? Now see what she does? She flips the slide over upside down and presses it onto the cover slip. And then flips it. So now if things go well, there will be a drop hanging down from that cover slip. Then we go over to the microscope and we go through the steps of how to focus a microscope. Remember, you bring the stage all the way up. Here's the edge of the drop. Here's the drop itself. Your best view is right at the edge. Now, this is very hard to see. It's not well focused. But in just a second, what we'll see is some cells. She's going up to a higher power lens now. This is the 40x lens. Again, we want to look right at the edge because that's where most of the cells are going to be. And if you look very carefully, and again, I apologize, it's not very good quality here. See, there's some movement right there. See a little bit of swimming around of cells. Very hard to see. If you can't see it, don't worry, trust me. There's some things swimming in here. And those things are right up against the edge. There's some right there swimming. There's one there. There's one there. One more time here. We're under the 40x lens because that is the minimum lens we need in order to see bacteria. See them swimming? That is the hanging drop technique. Any questions about that? Any questions about how that's made? The big difference, again, between the wet mount and the hanging drop is how you make them. The wet mount, you're putting a drop on a plain old glass slide, and then you're putting a cover slip on top of it. Very simple. For the hanging drop, we're putting a very small drop on the cover slip. We're going to put some Vaseline around a circular area on the slide, and then we're going to flip the slide over and press it down onto the cover slip, and then flip it back. So we put the drop on the cover slip, we take the slide, we press it down onto the cover slip, and then flip it back. And hopefully, if all goes well, the drop will be hanging from the cover slip and not touching the well. And we'll be able to see if we have any motile cells. Now, I mentioned at the beginning of the lab today that we have to be able to know when we see something moving on a wet mount or on any preparation like a hanging drop, when we're looking at live cells, we have to be able to know if the movement we're seeing is actual motility, true motility, or something called Brownian motion. Brownian motion. Now, Brownian motion can be a problem under the microscope because it can be difficult to tell whether you're looking at Brownian motion or true motility. All Brownian motion or Brownian movement is the movement that's associated with molecules that are in a liquid striking the cells that are in that liquid and causing them to bounce or shake or vibrate. In other words, in that liquid, there are going to be molecules that are too small for us to see. All molecules have inherent motion. Remember from physics, all things vibrate a little bit. And those vibrating molecules are going to bump into our cells sometimes and cause our cells to move, to bounce or vibrate or shake a little bit. This is not the cell moving under its own power. This is not motility. This is a movement that's caused by something bumping into the cell. Now, Brownian movement, this sort of false motility, is especially evident when we're under high power lenses. So the 40x and the 100x. Take a look at this short video. We're looking underneath the microscope at a wet mount. Now this happens to be a water sample. There's a lot of debris in here. All right, so all of this material that you can see here, this is just debris. These little fibers, this is just stuff that's in the water in addition to any bacteria or microbes. But I'm going to put this in motion. I know it's hard because the light source moves a little bit here, but I want you to look carefully and see if you can see anything moving in here. There are some very small, here's a large one. There are some things moving in here. Can everybody see that? There's something moving right there. Let me put it again, put it in motion again. There are things that are moving. Notice that they're all moving from left to right. They're all moving at the same rate, the same speed. There are bigger things moving. There are smaller things moving. See that? There are things moving in here. That is not motility. That's not even Brownian motion. What you're looking at here is literally a current in my wet mount. When we put a drop of fluid onto a slide and put a cover slip on it, that fluid can be in motion for several minutes. It's like a current traveling down a river. Now, how do I know that I'm looking at current and not motility or Brownian motion? Well, when it's a current, all of the things are going to be moving in the same direction at the same speed. There's something moving right there. All of the things are moving in the same direction at the same speed. It's not just cells that are moving, it's bits of debris moving as well. So big things, little things, black things, white things, they're all going to be traveling in the same direction at the same speed. That's just the current that's in the water, in that drop of water, carrying material with it. Okay? Now compare that with this. Again, take a minute to orient your eyes. I want you to look in here and see if you see any motion. I know the light is moving and that makes it a little hard. Look at these big cells right here. These are yeast cells. You see them moving at all? That one? Take a look at these cells right here. This little cluster of cells. Do you see them? They're bouncing a little bit. They're shaking. They're vibrating a little bit. Yeah? Something's going to come in view here that's also bouncing and shaking and vibrating. Look at right in this area. You can see something right here that's sort of moving. It's bouncing, it's vibrating. It's going this way. These are sort of staying in place. This thing is going to now travel this way a little bit. What you're looking at there is Brownian motion. You don't see that current effect. You don't see everything traveling across the slide. You see some things that are just kind of going like this. They're just kind of bouncing, vibrating. They're not really traveling forward particularly. They're just kind of vibrating in place. Let's look at it again. This is Brownian movement. The reason these things are bouncing around a little bit, let's get in focus here. The reason this little thing is bouncing around, vibrating in place, is because other things are bumping into it. Other things are bumping into the cell and this one. It's Brownian, it's not real. In other words, it's not that the cell or the particle is moving under its own power. Something's bumping into it and causing it to move. That's what we call Brownian movement. Here's another one. Look at these cells right here. There's 3 cells. These are big cells. They happen to be yeast. See how they're just sort of slowly going off to the left, and they're vibrating a little bit. They're not moving as quickly as they would if they were in a current. And the other reason I know it's not a current is because there are other things moving that are not going in that direction. There are other things that are moving around, vibrating, that are not traveling in this direction. There's something right there that's vibrating. That's Brownian movement. That's not real. So how do we know if we have real movement? Let's go back to our Enterobacter that we looked at earlier. When you have cells that are truly motile, they're going to be moving very obviously. And notice how they're all moving in different directions. See that? Some of them are going very, very fast. They change direction. They're all traveling in different directions at different speeds. It's very obvious when you have true motility, because you have cells that are traveling under their own power. So they're going in their own direction at their own speed. They're not paying attention to anything else. They're just off doing whatever they're going to do. That is motility. Very, very different from both that current movement and the Brownian movement. When you're first learning how to use a microscope, it's very easy to mistake the movement of a current or Brownian movement with actual motility. But once you've seen enough slides with motile cells, you start to notice the differences. Motile cells will really jump out at you because, again, they're moving much more obviously, much more quickly. They're going in all kinds of different directions. They're changing directions. Yeah, very, 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 very different sort of motion when you have mobile cells. All right. In the second part of the exercise today, what we're going to do for the rest of our time is we're going to make a wet mount and we're going to take a look at it. What you're looking at here is my hand, and I'm holding a plastic 15 ml single-use test tube. We use these plastic test tubes a lot in the microbiology lab. We can put some broth in here and we can grow microbes pretty easily. And then when we're done, we simply toss that tube into the autoclave, sterilize it, and then throw it out. Now, what you're looking at in here is a homemade culture. And I'll tell you how I made it. I took a strawberry that I bought at the grocery store and I mashed it up in a little bowl. And then I covered it and I left it out at room temperature for a couple of days. This is a tried and true way to grow some yeast. If you want to grow some yeast and look at them under the microscope, all you have to do is draw the liquid off from around the strawberry, put it into a tube, and then take a drop out of the middle of the tube and examine it as a wet mount. Now, here's a question. I bought this strawberry from the grocery store. I'll tell you, I rinsed off this strawberry like I would if I was going to eat it. I rinsed it off under the sink, and then I patted it dry, and then I put it in a bowl and I mashed it up. And a couple days later, as you're going to see, I grew a whole bunch of yeast. I grew some fungus. So the question is, where did that fungus come from? Where'd that fungus come from? How did I know that if I mashed up a strawberry and covered it with some tinfoil for a couple days, how did I know that I would get fungus? Any ideas? Any ideas at all? I'll give you a big hint. Humans are not the only organisms that have a microbiome. Lisa is talking about temperature and sugar. Lisa, you're exactly right on the sugar front. I mashed up that strawberry. So what I did was I released what was inside the strawberry. And I knew from previous experience that there is fungus on the surface of strawberries. There's yeasts on the surface of strawberries. Remember, strawberries grow outside and they grow close to the ground, close to the soil, right? And it's perfectly normal for there to be fungus on the surface of that strawberry. You wouldn't find fungus inside the strawberry, but on the surface of the strawberry, it's perfectly normal. It's part of its microbiome. And remember, you cannot wash off a microbiome. You do not wash off your microbiome when you wash your hands, even if you're washing with soap. You will wash off any free living cells, any planktonic cells that you might have picked up, but you won't wash off your microbiome because your microbiome is in some biofilm, right? Well, the same is true of a strawberry or an apple or a cucumber or cabbage or any other plant. They have their own microbiome. And yes, it's important to wash those things because you're going to wash off any free cells and you'll also wash off some pesticides. But yeah, if you mash up a strawberry, what you're doing is you're going to feed the yeast that are on the surface of the berry. You're going to feed them the sugar that's inside the berry. And fungi love sugar. Good job. So I made a wet mount using my homemade strawberry culture. Now notice I have gloves on because of course I'm handling microbial culture. It's not a dangerous culture, but I'm still wearing my gloves because I always use good technique. I do want you to notice something though. These gloves don't fit me. When you wear gloves in the laboratory, the glove should fit in a way that is close to your skin, but not tight. This glove is a little bit too big for me. All these wrinkles that you can see, there's a little extra room in the fingers. This glove is just a little bit too big for me. I would preferably use a glove that was one size smaller. You can really see it in this image. See how wrinkled those gloves are? Folded. This glove is too big for me. Now, I'm going to draw my sample out of this tube. I want to show you the correct way to open up a broth tube in the laboratory. Anytime you have liquid broth where you're growing microbes, when you open up the tube, you're going to want to hold the tube at an angle. Remember, microbes fall out of the air. They don't jump up off of surfaces, but they fall in air currents. So by keeping the tube at an angle, I'm making it less likely that something that is falling down will land in my tube. Okay. The other thing to notice is that I'm holding the cap in my hand. See how I've got it nestled between two of my fingers? Don't put the cap down on the surface. Because remember, we always assume that surfaces are contaminated. Even if we just disinfected, even if you just disinfected your worktop, you would never put a sterile cap down onto a workspace because then it would be contaminated. So when you take the cap off, you simply tuck it between your fingers and hold on to it while you're working. Draw your sample out and then close that tube back up again as soon as possible. And that way you'll reduce the chances of contaminating your culture. Now, I've used one of those plastic transfer pipettes here to put a nice big drop of strawberry culture onto a glass slide. Notice I've got my slide labeled at one end, of course, because we always label everything in the lab. Look at that good sized drop right in the center of the slide, about the middle third of the slide, right here, and right in the middle. This is a nice sized drop. I don't want the drop too big because then it's going to run out from underneath my cover slip. But when I'm making a wet mount, I can use a nice big drop. If I'm making a hanging drop, though, I want a small drop, basically the amount that's in a loop of culture. Now I'm putting my cover slip down. See how I put one side of the cover slip down first? That's going to draw the drop over to it by capillary action. And then I can slowly lower the other side of the cover slip and I'll get very few air bubbles underneath. Now, once I'm ready to take my slide over to the microscope, my gloves come off, right? We don't use gloves while we're using a microscope, because we assume our gloves are contaminated and we don't want to handle anything when we have gloves on that is not routinely disinfected. Microscopes are not routinely disinfected. So always take your gloves off before you examine what you've made. Any questions about what I did there? I took my culture, my homemade culture, my strawberry juice. I would have made sure to mix it up very well to resuspend any cells in there. I took a sterile transfer pipette, tilted the tube, took the cap off, put it right in my fingers, took my sterile pipette, put it into the center and drew up a drop. Cover my tube back up. Put the drop right onto the center of a microscope slide and put my cover slip on top. Sarah's asking a good question. Do you wear your gloves while you're placing the slide on the microscope and then remove your gloves? Sarah, that's a great question. And it's, you know what? It's going to depend on the person. I personally keep my gloves on until the slide is on the microscope. I personally don't handle slides without gloves on when I'm putting them on a microscope. As soon as the slide is on the microscope, my gloves come off. But I have seen plenty of people who will make their slides and then take their gloves off and then put the slide on. It's really personal preference. The important thing is that you don't want to be handling all the knobs on your microscope and the eyepieces and the lenses. You don't want to be handling those things with your gloves on. All right, so let's take a look. Let's see what I found underneath my slide. So this first image I put in because one of the things that sometimes we do when we're learning how to use a microscope and we're learning how to focus a microscope, when you're putting the drop of material onto the slide, sometimes we draw with a Sharpie pen, we draw a circle first. Take a Sharpie pen, draw a big black circle in the middle of the slide, and then put your drop of fluid right into that circle. The reason you would do that is because of this. When you're trying to find your drop, when you first look under the microscope, when you're trying to find your drop, you can look for the black circle. Look how big and obvious it is under the microscope. I'm under a low power lens here. I'm under a 4 or 5x lens. And this big black line is the circle that I might have drawn with a Sharpie pen. If you can find the line, you can then find your drop right next to it. So sometimes people draw with a Sharpie pen, draw a circle around their specimen on their slide, just to help them find their specimen when they're first learning. So here's our specimen. We are under a 4x lens, a low power lens. And remember, that's a 40x total magnification because I take the power of the lens, that's 4x, and I multiply it by the eyepieces, which are 10x. 4 times 10 is 40. I have 40x total magnification here. Now, one of the first things you're going to see here under this field is this blue thing. What the heck is this? It's blue. I didn't put any stain on this slide. What the heck is this? Well, this is just a piece of debris. This is something that was either on the strawberry or perhaps in the dish that I used. It's just some piece of debris, microscopic debris that got into my sample. Everything else you see on here in brown, this is fungus. So all of these branching structures that looks like a tree almost, looks like these little trees growing in here. This is fungus. And in particular, this is mold. This is what we call mold. We're going to talk about fungus. We're going to spend a whole lab talking about fungus in a couple of weeks. But for today, understand that fungus in its microscopic form, the kind that we would call microbial. Remember, a microbe is microscopic, single-celled, right? Either a cell or a particle. That's a microbe. The microbial form of fungus is called yeast. Now, a yeast is a eukaryotic organism. Fungi are eukaryotes. They're not bacteria. They're not archaea. They're eukaryotes, eukarya. And the microscopic form of fungus is called yeast. Now, there's another form that fungus can take that can be either microscopic or macroscopic, and that's called mold. So yeast is the microscopic form of fungus. Mold can be either microscopic or macroscopic. In other words, you might need a microscope to see it, or you might not. It depends. Depends on the species of fungus. So when you hear people talking about mold, it's important that you know that that is not a type of fungus. Mold is not a type of fungus. It's not a species of fungus. Mold is a form that fungus can take on when it's growing. So every species of fungus can grow as a mold. Molds can be molds are multicellular. So they don't meet our definition for a microbe. Yes, they can be microscopic, but they have multiple cells. So they don't meet our definition. Yeasts meet our definition. They're microscopic and they're single-celled. But molds don't. They can be microscopic, but they have multiple cells. They look like trees. Molds look like trees when they're growing. They have these branches that grow out. Now, under this low power lens, I can clearly see those branches. I can clearly see these, the trees. The question is, do I have any yeasts under here? I'm going to have to go to a higher power lens to figure that out. We're looking under the 100x lens now, the oil immersion lens. Let me come out a little bit. So remember, that's 1000x total magnification. 100 times 10 is 1000 total magnification. Here's the mold. Here's a branch of mold right here. See the branches coming off of it? Branching mold. Here's yeast right here. Yeast cells grow off the ends of the branches. They actually bud. They bud off of the branches. So here's a full, complete adult yeast cell. And here's another one that is now budding off of the first one. In this particular species of fungus that tends to grow on strawberries anyway, the yeast will start to come off of the ends of the branches and it forms long chains. It's kind of cool. I can't tell you the name of the species because I don't know it. But look at this chain of cells coming off of that branch. This cell developed first, and then it budded a new cell. And then another one budded off of it. And then another one budded off of it. And it keeps going until you get these long chains of circular cells coming off of these branches. Now, eventually, these yeast cells will break off. They'll break off and they'll go somewhere and they'll grow a new organism, a new mold. Fungi are fascinating, absolutely fascinating. Look at this view. We're right at the edge here of the field under the microscope. We're still under the 100x lens. You can see the tips of some of the branches of the mold, and you can see some yeasts budding off. See how big the yeast are under the 100x lens? Oh, sorry, let me go back. See how big these circles are? Much, much bigger than the bacteria that we saw under the 100x lens. Much bigger. These are fungi. They are eukaryotic. They're much bigger than prokaryotic cells. These black dots here that you can see, these might be bacteria. Bacterial cells are much smaller than yeast cells or animal cells or plant cells. They're much smaller. Down here, what we're looking at is a different field of view on my strawberry sample under the 40x lens. Let's take a look at this. In this area, you can't see any trees, but you can see these things right here. See these like worm-like things? See those? See those? Over here, same thing. Some little wormy-like creatures in here. Here's a question. Do you think that these are bacteria? Could these be spirochetes or spirilla, these cells? Remember, spirilla and spirochetes are the sort of wavy looking coiled bacteria. Do you think that's what those might be? Why or why not? Remember, bacteria can be cocci. Those are the circles or the spheres. They can be rods. Those are the elongated ones. They can be spirilla. Those are the sort of twisty ones. What do you think? Candy's got it. Lisa's got it. Brooke's got it. Maddie's got it. Good. Very good. No, not bacteria. Too big. Too big. We're under the 40x lens. We're under the high dry lens. You can see bacteria under this lens. You can see them, but they're going to be very small. What we're looking at here is much too big to be bacteria. It turns out that what it is little fragments that have broken off of the mold. Just little pieces of the mold, the tree, that have broken off. That's what those elongated things are. Can you see some circles in here as well? This one right here, see that black circle right there? There's another one right here. There's some up here, very circular looking. Those are yeast. Those are the yeasts. Again, we're under 40x. Bacteria would just be visible as little pinpoints under the 40x lens. This is a yeast cell right here. Here's another yeast cell. Here's a piece of the mold. Here's a piece of the mold. Think about your magnification. Think about your magnification before you decide what you're looking at. Now, on this slide, this is a wet mount that we're looking at. All I want you to look at on this slide are these things. Does anybody know what these white things are? They're all different sizes. They're perfectly round. They have a dark ring around them. Anybody have any idea what those are? I made a wet mount, put a drop on a slide, put a cover slip on it. This one happens to also have some color in it. Anybody know what those are? They're always white. They always have a dark ring. They're always perfectly round, and they come in different sizes. Maddie's got it. Brooke's got it. Amy's got it. Lisa's got it. Air. Those are air bubbles. Those are air bubbles. They always look like that. Look for that dark ring. Perfectly circular, white, with a dark ring. That's an air bubble. You just got a little air underneath your cover slip if you see those. No big deal. You just need to know what it is. I did a little trick here. I did a little trick on this slide. I took my wet mount with my cover slip, and in order to make the air bubbles stick out a little more, I didn't use a blue filter. But instead, what I did was I took a drop of blue stain and I put it right at the edge of my cover slip. When you put a cover slip down on top of a wet mount, if you put a drop of stain at the edge of the cover slip, it'll get drawn under by capillary action. So again, I did it here just so you could see the air bubbles a little more clearly. Remember, stain will kill anything that's living in here, so don't put stain on until you're ready to kill whatever microbes are there. Here's our strawberry sample under the 4x lens. See, I've got a sort of tree formation here, a branching mold structure with little yeast cells coming off. This is 4x. Look at the size of the yeast cells under this low power lens. I would not be able to see bacteria here, but I can see fungal cells. I can see yeast cells. Here's the same image, the same piece of that organism, but at the next power up. This is the 10x lens. See the yeast, you can see them in much more detail now. You know why? Because I put the stain in. I added a drop of stain. Look how much more detail you get. We use a lot of stain in microbiology because of this reason. It's a lot easier to look at these things when I add the stain in. It just gives it more detail. I can see branches of mold and yeast coming off of them. Now, the next time we get together for lab, we're going to talk about what's called simple staining. And all simple staining is making a microscope slide that has bacteria on it that we apply a simple stain to, one stain. We're going to talk about that on Monday. And then on Wednesday next week, we're going to talk about a different kind of staining called gram staining. So next week in lab will be all about staining. And I'll show you why. I'll show you the benefits that we get when we stain cells, how we can see them just better under the microscope. So that'll be next week. You're all set now. You're all done now with your lab material for the week. So you can go do your lab questions anytime now for this week. Remember, you have a lecture exam. You're going to need to take that sometime between tomorrow morning and Saturday evening. So whenever your work schedule, your life schedule permits it, you'll need to sit down and take the exam. Once you start it, you need to finish it. Okay? Once you start the exam, you're going to need to finish it and submit it. You have 90 minutes for full credit. Most students, again, take about half an hour to an hour to finish this exam. Remember, you have to go through Respondus. You have to use the lockdown browser. And you can expect between 25 and 30 questions. Okay. You have a quiz due tonight at midnight. That's your cells, the cells lecture. You have a quiz tonight to do by tonight. You have lab questions to do by Sunday. And you have that exam to finish. Okay. Any questions about what we talked about today? Any questions about the wet mount or the hanging drop and why we would do that? Why would we bother? Why would we make a hanging drop? All right. All right, very good. Now listen, if any questions come up between now and Monday when I see you again, don't hesitate to message me, okay? Message me through Canvas if you have any questions at all. And certainly, if something goes wrong, if you have something come up and you can't take the exam, message me immediately, okay? Or if the power goes out and you have no Wi-Fi, message me, okay? Let me know. We'll figure it out, all right? Remember, you're not allowed to panic in this class. Don't ever panic, okay? We'll figure it out if something goes wrong. All right, everybody, I'm going to let you go. Enjoy the rest of the day. And I will see you again on Monday morning. All right, everybody. You're welcome. You're welcome. Take care.